Now, this chapter is mainly going to focus on the cell and different components that are present within the cell. The word cell is a term that you can associate with something that is enclosed and has boundaries or a wall around it, or some sort of a barrier around an enclosed space is what the term cell indicates. Cells are the fundamental building blocks of living organisms. They are simple organisms such as unicellular organisms, which is an amoeba or a euglena or a chlamydomonas or a bacteria. These are just one cell organisms, and the cells can then be advanced into more multicellular organisms such as a plant or a mouse or a cat or a flower or us as humans. So keeping that in mind, let's look at the different parts of a cell. The cell always will have a plasma membrane. A plasma membrane is like your wall that you can think around a room or around a house. So it's the outermost boundary that would be present in all cells unless they are plant cells. So if I was to draw a region and say this was the boundary where everything is going to be enclosed within the cell, within this region, then this would be my plasma membrane. Now within the plasma membrane are fluid and organelles or structures that are suspended in this fluid. The fluid of the cell is called as the cytosol, and the structures or organelles could be present within this cytosol. Together, all of this is called as the cytoplasm. So in other words, cytoplasm is going to have a fluid part and also the structures on it. So if you think of your room, then the floor itself would be the cytosol, and all your furniture and your equipment together would be the structures and the organelles. Within the cell, some cells that are eukaryotic cells have a nucleus, and we'll give you a little bit of hint later on what is the difference between eukaryotic and prokaryotic, but the central region in here is the nucleus. You can see the same nucleus as over here, and you can see the nucleus over here. So these I've pointed out to the central region within, actually in this, or we would consider this whole region to be a nucleus, not just the dark region. However, in cells that are prokaryotic, which means that these are cells that do not have any compartmentalization within the cell. So in a prokaryotic cell, all the structures would be present floating or within the cytoplasm. You may have granules, you may have different parts that could do different functions of the cell. The eukaryotic, on the other hand, have very strict regions that are either called as the chloroplast or a mitochondria, or it might have a membrane system, and the nucleus is also more membrane bound. So a membrane bound is a eukaryotic cell. So eukaryotic cells would have membrane bound structures. Prokaryotic cells would just have a cell membrane and everything would be suspended in there. Now, another interesting thing is when an organism grows in size, the surface to volume ratio is very important. So, for example, if you try to cook a large piece of steak versus, let's say, a 5 pound of a roast beef or a large piece of steak, and you have ground beef about the same weight, you know that the ground beef will, with experience, cook faster. Why? Because it has more surface to volume ratio. So, if the cell was to get from a small little size to get enlarged, it would not have the same surface to volume ratio if, on the other hand, the cell was to make many copies of it and occupy the same amount of space. Then the surface to volume ratio will increase when there is a multicellular organization. So, keeping that in mind, let's look at the term which is called as the cell theory. Cell theory is a very old-fashioned concept about how the term cell came about. The first statement is that organisms consist of one or more cells. So as I said, either prokaryotic or eukaryotic, they have to have a basis or a foundation of this of the building block. Even the smallest unit will retain the capacity for life. So for instance, the smallest unit of a bacterial cell will also have a capacity for its growth, its metabolism, carrying out its function, its cell reproduction, cell division, response to stimulus, and so forth. And new cells will only arise from the growth and division of precursor cells. This is very important, because if we did not believe in this precursor of a cell, then cells could just randomly be formed by just a magic wand, or from inorganic substances, or from a rock, or from dirt. But that is not really true. Time has proven that many experiments were done to prove that cells usually arise from pre-existing cells. Now on this part, and in this image over here, I have a scale that depicts how small a tiny cell is. So if you compare yourself as humans in here, and then you look at a cell, its size of this this organism, or with the human eye, you can see that. You can see with the human eye, you can see a bird. You can even see a small egg, but you cannot see a cell with a human eye. You need a light microscope to show you the size of a cell. And in another more fancier microscope is called as electron microscopes, which has a dimension of in nanometers, or sometimes in microns, and you can see structures within the cell. So when you see with a light microscope, you will see just structures within a cell, but you really cannot recognize each region. Now, the reason I'm pointing it out to you is because when you look in the books and you look online and you see a lot of nice pictures, a real image of a cell within a light microscope is no more closer to it. Electron microscopes cost thousands of dollars, and the publishers have paid good amount of money to get these images embedded in their books. So when we draw a cell, we have a very crude picture, and when we look at it in a microscope, we don't see all the details, but studies have shown that there are these structures present within the cell. So coming back to how do we see these cells, as I just mentioned, the cells would be seen with an equipment that is called as a microscope. Microscope pretty much magnifies a small section of a sample and enlarges it depending on the zoom power that you have and depending on the magnification of the lenses that are used. And there is a assigned lab in which you will learn about the different parts of a microscope and how this works. So I'm not going to go into too much depth here because you have a task to do that. Quickly, I want to go over that either a microscope works with a light, which is called as a light microscope, because light is passed through a very transparent object, and it magnifies the object through lenses to reveal a more clear image. However, if you want a better penetration, then electrons are used to penetrate an object, and you will have a much sharper and more detailed image.